What is then that value, that unique value and added value of the BIACOR platform in drug discovery? It's really, as I mentioned, the capability of getting kinetic information of an interaction. If you're looking at these three blue dots over here, they have the same affinity, around 10 nanomolars. And if you were using an equilibrium-based method, you would say that, well, they have a similar binding behavior, they have same strength of binding, so I could take either one of them. But in fact, if you resolve that affinity into kinetic parameters, the on rate and the off rate, then you can really look into characteristics of binding that can be very important from a clinical perspective. So, for example, if you have a very rapid on and rapid off rate, maybe you need to administer a drug in its formulation at low dose with frequent administrations. On the other hand, if you have a slow dissociation rate and a long residence time of your drug, you may need just one single or two single administrations to get the same kind of effect. And that can be desired from various perspectives, not only the convenience for the patient. And there is a nice publication which I can recommend from Dr. Robert Copeland, the former vice president of JSK Discovery. He published a paper around the correlation between target residence time and in vitro efficacy based on kinetic parameters like dissociation rate. If we're looking at label-based approaches and label-free approaches, how can then the label-free approach really meet the current challenges and trends in drug discovery? One of the trends is, of course, fragment-based drug discovery now and structural-based approaches, where you are actually looking at alternatives or complementary ways of supporting your high-throughput screening. And here we are starting to work with lower molecular weight compounds, small chemical probes or fragments below 300 Daltons. Well, that really requires a very sensitive technology, and the BIACOR technology, for example, is really has a capability of detecting those low affinity interactions and monitoring those in real time. Another important thing with the label-free technology is that you can work with problem targets. For example, an intractable drug target or a target where you have an unstable substrate or where you may not even know the substrate of interest, so that you really cannot address that with a biochemical screening assay. And other things, I think, is just the cost issues and the possibilities of working with very low volumes and amounts of protein that you can do in the label-free technologies like the BIACOR platform. Let's have a look at some real industry data. This data I'm going to show you now is from the spectroscopy group in Basel in Switzerland, in Hoffmann-La Roche. It's Dr. Walter Huber's laboratory. And they are actually following up the HIT series from the HTS based on the SBR, the BIACOR technology. In this case here, it's what he calls an SBBA approach, a structural-based biophysical approach, where they are using a pharmacophoric model and in silico screening to find structural classes of interest that may be interesting for a specific target. In this case here, it's a target called HPPK, a kinase target, involved in a disease process. So what they will want to do here is to study the interactions using SBR. And the reason why they chose in that type of approach here is that the HTS didn't really work because of the unstable substrate in this specific process. So using in silico screening, they selected 250 compounds based on that structural data. And that represented about 10 classes. And another thing you can do here as well is the specificity of binding, the selectivity of binding, and they actually made point mutations at the binding site of that therapeutic target protein, so that they actually have on one flow cell they have the wild type kinase, and at the other flow cell they have the impaired mutant of that target, so they can quite rapidly weed out those false positives. Interestingly, if you're looking at the HTS at Roche still today, about 80% of the hits coming out from HTS are in fact false positives. And it can be extremely expensive to continue with medicinal chemistry on false positive hits, which may be due to compounds forming micelles. It may also be due to other aggregation or fluorescence quenching, etc. So this is actually a very effective approach for them to move forward. screening the compounds of interest, and in this particular case, they could quite rapidly find out some real binders. What you see here is the wild type response minus the response to the binding site mutant. And you can quite quickly find the real binders here. And interestingly, if you're looking at this compound number 5 here, in the first approaches where they attempted some other methods, this was the highest responder, and it's really showing up not as a real binder in this case here. Another important thing in this experiment that was found was that the affinity actually doesn't really correlate with the selectivity in the end. And that can be quite important findings when you're selecting your starting point moving forward. So in this particular case, they found 4 good starting points, which were good starting points for further chemical synthesis. And it showed just a way to move forward when you don't have a stable substrate and can't address it from an HTS point of view. An alternative or a way to bypass the HTS process in this case. And in this particular case, it really saved the project, and it moved over to clinical phases in the end. So it was just fun to show with one example where it's not always fail fast, fail cheap, but also a way of saving projects in drug discovery. After finding your good binders, your true binders, whether you're working with a focused library or a fragment library or just following up on HTS, you may want to look in more detail during your hit and lead selection to the binding properties. And this is an example from another company in the US, it's Merck at West Point, where they have implemented kinetics as part of the selection criteria moving compounds forward. This is an example presented at a former SBS meeting from Dr. Delphine Collin. Just to illustrate here, these 2 compounds here have the same affinity but different dissociation kinetics. And in fact, this compound here has 35 times longer half-life. So what they're saying is basically they find that the best in vitro parameter predicting in vivo efficacy is the off rate in this particular case. It's not always the case, but it's a good prediction tool for making a good informed selection to increase the probability in later stages. And there have also been shown to be correlations to cell-based potency, looking at the dissociation kinetics. So I think this is an extremely important parameters, and more medicinal chemists are recognizing the value of this data. So this is really, I think, a big step forward for the industry in order to address attrition. So this type of technology has been implemented over a wide range of targets from these particular purposes. And I think what we've seen today over the past 10 years, SBR has really moved from a nice to have to a must have for many companies, where they have designed it into their processes. And over the next few years, I suspect we will also see a drift over to the ADME departments with this technology to try to combine more binding data with cell-based toxicity assays to front-load toxicity testing and look at panels of targets from animal models in order to improve the efficacy in later stages. So, does this concept also work for biotherapeutics and vaccines? Well, there are a number of good references, and I think we have about 10,000 of those in our database today, and I would say 30-35% of those references are around antibodies. So this is a technology that has been exploring antibody discovery and development for quite some time now. I would like to show and share one example from former Abgenix, now part of Amgen. This is a collaboration between Dr. David Mishka at the University of Utah and Scott Claycamp, former Abgenix. In this case here, they are screening hybridomas using the SBR technology to pick out binders to an antigen. And this is what you're seeing here. You have a wide range of affinities from 62 to 1 nanomolars for these 16 monoclonal antibodies. So first they are screening based on responses, and then they are picking out and purifying the binders, the best responders, and then injecting concentration series of the antigen, and that's actually the different blue curves here. From that, and this is the main point of the slide, you can present, again, an off-rate, on-rate map and look at, in this case, monoclonal antibody number 1 and 2, just showing that they have the same similar affinity but tenfold different binding kinetics. And this can be clinically relevant properties if you really want to optimize the efficacy in later stages, as also seen with small molecule compounds. So just to show you one example here from the antibody discovery. Now when we have selected our antibody of interest, we need to put that into the patient, because in the end we need to check what happens in vivo in order to make the most informed decisions on which candidate to proceed with. So in my following slides, I would like to discuss safety assessment from the point of view of clinical immunogenicity testing. And for those of you who are involved in biologics discovery and development, this is a key issue, looking at anti-drug antibodies in patient serum samples. There's, I think, one really good or one scary example from Amgen looking at the EPO drug that is still a blockbuster on the market. There was a few years back when patients treated with EPO here in Europe started to show some very critical side effects. We had patients with condition of PRCA, pure red cell aplasia, based on cross-reactive antidrug antibodies in those patients, neutralizing the endogenous EPO in those patients. And today the FDA and other regulatory authorities really require companies to monitor patient safety from an immunogenicity point of view and detect any type of immune response and then throughout the development monitor that immune response and the progression in each individual in the trial. So we are now in the space of preclinical clinical testing, and the example I would like to show you is from Boehringer Ingelheim in Biberach in Germany, where they are using both ELISAs and the BIACO platform to monitor the patient safety.